I obviously am going to be talking about voltage-gated calcium channels, and I do want to start by letting you know that a bunch of people from my lab are here, and so I'd love you to just connect with them. I can see Mary and Remy and Martine and Dimitri somewhere, and then Arturo Andrade, who did a lot of the foundational work that I'll talk about. He has his own lab, and he's here as well. So I'm glad that everybody has been able to come. So I think that you all well know that there's a large number of voltage-gated calcium ion channels, and there's a few that are dominantly expressed in presynaptic nerve endings. And the focus of what I'll be talking about, and I think a few other people will be, is the role of voltage-gated calcium channels in transducing information about nociceptive, transducing information that drives the nociceptive responses. But as I was thinking about what I would choose to talk about in the 20 minutes that I have, I'm not going to do justice to one piece of work, but I'm going to just kind of jump in and describe it very briefly. But then I've decided to really focus on the work that we're doing, looking at not calcium channels in the nerve endings in the dorsal horn of the spinal cord, but rather calcium channels in the skin, driving a hypersensitivity response. And I'll talk about some unpublished work as well. But I'm going to give a little bit of an introduction before I jump to that. So there are a number of different voltage-gated calcium ion channels, and they have different activation thresholds as well as expressed in different parts of the nervous system and, in fact, all other organs as well. And the channel that my lab has focused a lot on, it's not labeled here, but it's a calcium channel that is expressed in presynaptic nerve endings and certainly is expressed in the presynaptic nerve termini of nociceptors in the dorsal horn of the spinal cord. We call it CAV 2.2, or it's also known as alpha 1b. It doesn't really matter, but it's expressed at presynaptic nerve endings. And in nociceptors, it drives calcium entry that triggers the release of glutamate and peptides. So here I'm showing you actually the localization of these heat-sensing nociceptors in the dorsal horn of the spinal cord, and through a reporter channel rhodopsin 2 EYFP that's driven by the TRPV1 gene. So these are the TRPV1 nerve endings in the dorsal horn of the spinal cord. And then, of course, our lab and many other labs utilize optogenetic methods, and so we can also drive activation through using remote light sensing activation of TRPV1 nerve endings and get some really beautiful light intensity dose response curves so that the animal will withdraw its paw in response to light. So we can use that for our behavioral assays sometimes. And then ideally as well, this is a really good methodology to be able to look at postsynaptic currents just selectively driving certain sensory inputs. And in this case, it's the heat sensing afferents that are in the dorsal horn of the spinal cord. Okay. And the channel we work on, 2.2 channel, if we compare wild type and knockout, you can see that there's a right shift in the dose response curves to light intensity. And so this 2.2 channel is really important for driving baseline nociception. It's, move forward, here we go. Is that right? Oh, sorry. Right. We'll get there. There we go. And so these are the two very recently published papers from the lab. And I'm going to talk super quickly about this. This is work that Javier Lopez Soto did together. And this was really describing the molecular mechanism that drives cell-specific splicing of the gene that we're working on, the CAV 2.2 encoding gene, CACNA1B. And this is actually a very beautiful piece of work, and so I'd love to talk about it a little later if people are interested. But here's a summary, just very, very, very quickly. In the nerve endings of the TRPV1 expressing nociceptor termini, then there's, as you well know, calcium channels are modulated by a number of different G-protein coupled receptors, and in the nerve endings, in the dorsal horn, the calcium channel is downregulated by activation of the mu-opioid receptor, and that, of course, is a really important mechanism of its analgesic effects outside of the brain, of course. And there are two different forms of the N-type calcium channel generated by alternative splicing. And it's really interesting because the TRPV1 nociceptors dominantly express this particular isoform, and that drives a very prominent, large kind of higher level of inhibition through the mu-opioid receptor compared to the more dominant form of the calcium channel expressed throughout the rest of the nervous system. And the expression of this isoform is downregulated after nerve injury, and coincident with a loss or at least a reduction of morphine efficacy. So there's a relevance to the splicing mechanism, and what Javier did over the course of a few years in my lab is to really define the molecular mechanisms that are driving cell-specific splicing, and it turns out to be a cell-specific epigenetic regulation within the exon itself. That's all I'm going to say about that set of work, because what I decided to do is to focus on the other end of the DRG in the skin, and I'd actually love to know what Stefan thinks of this, and because now I'm going to be embarrassed because I'm going to be talking about immune cells, and I've got one immune cell in all my cartoons, and so I'm not going to say too much more about that. But we're really, really interested in thinking a lot more about the neuronal immune cell interaction in the skin, and so I'm really looking forward to getting input from all of you. So here's the skin. This is now the nerve endings that are the TRPV1 expressing nerve endings, and here we're driving TD tomato as a marker within the nerve endings in the skin. And the TRPV1 overlays with the CGRP expressing neurons, and so these are peptidergic TRPV1 expressing neurons in the skin. And this is a more recent 2P imaging in vivo. The nerve endings here in red, and this is showing you the blood vessels in green, and you can see how beautifully interspersed the nerve endings and the blood vessels are within the skin, and so we've been doing two photon imaging and measuring calcium signals within the nerve endings in the skin in vivo. And again, I'm not going to say too much about that, but Remy Meyer, who's here, she's going to be working on that in my lab. And we published a little bit of that in Dan DeBrule and Javier and Remy's paper just recently in Journal of Neuroscience. So I'm going to give you the kind of the end of my talk right here at the beginning, and then I'm going to provide some data to kind of back up the model that we're working on right now. So what we've been really primarily focused on is the TRPV1 expressing nociceptors. And so just to reiterate, we're really probing the nerve endings in the skin. And it turns out that what we've shown very conclusively now is that there are voltage-gated calcium channels in nerve endings in the skin, and these are the 2.2 subtype of voltage-gated calcium channels that are also expressed in the presynaptic nerve endings. And when the TRPV1 receptor is activated, for example, by capsaicin, and if we believe if the voltage-gated calcium channels coincidentally activated, so there's a convergence of two calcium sources, there's a release of neurotransmitter from the nerve endings. We know ATP is released, and I'll give you evidence for that. Certainly, substance P and CGRP are also likely to be released. And there's a feedback, of which we're trying to work out right now, and I'll show you some of our preliminary data on that Mary Saliba's been working on. And that feedback mechanism drives the hypersensitivity response in the nerve endings, so that now a lower temperature is going to drive activation of those nerve endings. And so you get heat hypersensitivity. And so I'll show you a bunch of data that we have on that. What's very interesting is that when we block the 2.2 channel in the skin, I'll show you the data, we don't affect the mechanical hypersensitivity response to the same stimulus. So we've been able to separate the heat hypersensitivity from the mechanical hypersensitivity using a calcium channel 2.2 blocker, conotoxin. And so again, I'll show you that data. So the specificity. There's really beautiful selectivity for this hypersensitization response in the skin. And you'll see that there's no impact on baseline nociception response. So it seems to be highly selective for this hypersensitization response in the skin. So I'll show you this data So here, this was the first thing we did, and this was done by a former graduate student in the lab, Dan de Broel, together with Javier. And this was actually a control experiment way back when we were doing these experiments. And so this is looking at response latency to a heat stimulus on the hind paw of the animal. Oh, shown over here very nicely. And so we actually were, in this experiment, it doesn't really matter, but it's kind of interesting for students in the room. We were really interested in probing the presynaptic nerve endings in the spinal cord, and we had to do a few control experiments. We didn't think that this was gonna, we had no idea that we were gonna get this outcome, but this is the control experiment. So this is like saline injection saline injection, a bunch of animals, and then capsaicin injection, and you can see the time scale here. This is a really robust hypersensitization to a heat stimulus applied to the hind paw. Within 15 minutes, there's a really strong hypersensitization, and so the animals are moving their paw away on order of about 3 seconds compared to about 18 seconds in the control condition, and then the response recovers really rapidly within 30 minutes. And this is the total, a number of different animals over here showing you the en masse data that we have. And so hopefully in the next slide, yes. And then we looked at the knockout animals. So this is global knockout. So I'm going to get to local block in a second. This is a global 2.2 knockout animal. And you can see that there's an increase in the baseline, and that's because of loss of the calcium channels in the presynaptic nerve endings in the dorsal horn of the spinal cord. Okay, so there's a change in the baseline responsiveness. But what we were really stunned by was this decrease in the response, the decrease in the hypersensitivity response. And you can again see the average data over here, and we weren't expecting that. And so then we started wondering whether, in fact, this might have something to do with, calcium channels in the nerve endings of the skin. This is just a control experiment that we were asked to do for the paper, and it's a good one, which is like, well, is there any change in the capsaicin responsiveness of the cells in your control and knockout? No, there isn't. When we looked at mechanical hypersensitivity, which is shown here, there was no impact. The knockout animals were just as responsive and showed just the same level of mechanical hypersensitivity as the wild type. And so that's shown here, and you see the same change in the baseline responsiveness, but the degree of hypersensitization is the same in the wild type and the knockout. And so then we did a series of, Dan did a series of experiments, and as did Javier, I'm sorry, so they both did these experiments. And we thought, okay, well, let's do local block. Let's just inject conotoxin into the skin and then do the test again, because this was a global knockout, and so we weren't able to pinpoint that the effect was coming from the skin. And so I'm just looking at my time here. Okay, good. And so that's the experiment here, where they just actually, they actually co-injected conotoxin with capsaicin right directly into the hind paw of the mouse. And lo and behold, you can see that this is a control, and then here's the reduction with conotoxin co-injected with capsaicin. And so it didn't completely obliterate the hypersensitivity, but it was significantly reduced. Dan went on and showed that when we inject conotoxin directly in the skin, there's no effect on baseline nociception, which we weren't expecting. And then the other experiment, which is also very beautiful, and Remy did this, showing that if we use a 2.1 blocker agotoxin rather than a selective 2.2 blocker, there's absolutely no effect on the heat hypersensitivity. So highly selective for 2.2, not 2.1, highly selective for heat hypersensitivity and non-mechanical. So then we were like, well, okay, so if you don't activate the TRPV1 receptor, you don't see hypersensitization. But if you block the 2.2 channel in the periphery, you see a very reduced hypersensitivity reaction to heat. So we thought, well, it's likely that something's been released, and so we actually had access to we tried a few different things, but found that a blocker of the P2X7 receptor was also effective at reducing the hypersensitivity response. So we can rule in ATP as a key signaling molecule, initial pro-inflammatory mediator of this response. We can't rule out anything else because we haven't probed that sufficiently well yet. But we can certainly rule in ATP as a key player. And so this is control response. This is capsaicin together with the P2X7 receptor inhibitor, which is not expressed in neurons and only expressed in non-neuronal cells, including immune cells. So that pulls in the likelihood of a signal going to immune cells and then something else being released. And so what is that something else? And so this is where Mary Salib has been working in the lab, trying to define the secondary immune signaling molecules that might be driving then the hypersensitization of the TRPV1 expressing neurons. And so Mary's been doing a lot of really beautiful work defining the inflammasome in collaboration with Amanda Jameson, who's an immunologist at Brown. And so we've been working with her lab. And remember, I just want to kind of remind you, this is a very, very, very rapid response. So within 15 minutes. And so Mary kind of used a number of different panels and identified IL-1 alpha as being elevated in response to capsaicin. She doesn't see any evidence for IL-1 beta being elevated right now in this very short time frame. But if I have time, I'll show you some data using CFA. And there's a very similar pattern of importance of 2.2 in driving the CFA much more prolonged response. But for now, I'm going to show you this. And so she's done a number of different experiments. So one is to look at IL-1 alpha levels in the fluid extracted from the hind paw within 5 minutes of doing the capsaicin injection. And you can see here, compared to saline injection in the hind paw fluid, there's a higher level of IL-1 alpha following capsaicin injection. So that's taking directly the fluid from the hind paw. And then following on from something that Dan did, and that I think is almost certainly in the Journal of Neuroscience publication, although I think we had it out and then we were asked to put it back in. But this was kind of a really neat experiment where he took skin extracts and then incubated the skin extracts overnight to see is there anything released from into the conditioned media, and in fact, IL-1 alpha is also released found in the conditioned media, and that's reduced in the calcium channel knockout mice compared to wild type. So that's also kind of an interesting observation, suggesting that 2.2 channels are important in also driving the release of basal level of IL-1 alpha. And then this is, I think, a real key important experiment that Mary did, which is to then do an absorption experiment to neutralize IL-1 alpha in the hind paw of the animal to see if you neutralize IL-1 alpha, does that have an impact on the hypersensitivity response that she sees after injection of capsaicin? And in fact, yes. So this is actually, I think, really nice. Like you see it, then you block it. And this is the block, the neutralizing experiment. And so this is a classic capsaicin response to capsaicin response, hypersensitization of the heat stimulus within 15 minutes. And then this is the response to capsaicin plus the neutralizing antibody. And you can see that's not different from the control experiment, which is really great. So that suggests, very, very, very strongly suggests that IL-1 alpha is playing a really key role in this heat hypersensitivity response. So this is our model then that we're working now to measure the calcium signals in vivo using 2-photon imaging to get a sense of what's the kind of the critical threshold calcium that's going to be required in order to drive this hypersensitivity response. And that's where we've had to build a number of optogenetic models because that's actually easier in the 2-photon imaging than poking animals or heating the paw of the animal. It's much easier to be able to use the light activation. And so that's what Remy's spending a lot of time doing. But the model, just to go back to the model, is that we believe that there's a key kind of threshold amount of calcium that's required in order to drive exocytosis of neurotransmitter, and we believe that the 2.2 channel is absolutely critical in driving this heat hypersensitivity response, but not mechanical. Okay. And finally, I think I do have a little bit of extra, yeah. Okay, I'll do it super quickly just to show you the rest of the unpublished data to get your feedback later. But that's a very, very, very brief response, capsaicin. But of course, there are other ways to drive an inflammatory response that can last for days, and adding CFA, complete Freud adjuvant, drives this very, very, very long-lasting and profound hypersensitization. And so you can see here contralateral and ipsilateral paw, and this is post-injection of CFA. Okay. Oh, this didn't work out well. They just did it. Anyway, this is N of 12, but this is contra and ipsi, and this is in the knockout compared to wild type. That's stunning. So there's a really, really strong dependence of 2.2 channel also on the CFA, very prolonged hypersensitivity response. And that is also specific for heat hypersensitivity, not for mechanical allodynia. So again, like that's really showing that there's this divergence of these two pathways. I mean, of course, different mechanisms, but I think very beautifully showing one depends on 2.2, the other does not depend on the activation of 2.2 channels. And then just as I showed you before, well, this is the global knockout, but what if we inject conotoxin directly into the skin? And I think this is a really powerful experiment that Mary did, showing that, so here's the CFA only, here's contra and ipsi, but here's the contra and ipsi with conotoxin injected directly into the skin with CFA. So this suggests that just a kind of a brief exposure to conotoxin at the right time is able to really, really reduce this heat hypersensitivity response to CFA. So that's, I already did the summary. I talked a little bit about this, and then I'll just end with the cycle. So we're really, really, really excited about this, and we're really interested in probing this further. What's the critical calcium concentration required to drive hypersensitivity? What's driving hypersensitivity in the mechanical nerve ending pathway, and Remy is doing a lot of work on that right now as well. And so we've generated a number of different animal models for that, and I can tell you a little bit about that if you're interested. But I think really an important and kind of unappreciated role for voltage-gated calcium channels in the periphery, driving this hypersensitivity response. And as I'm going to leave you with the photos of the key people who did the work, I mentioned Javier, I mentioned Dan, Arturo Andrade did a lot of the really important kind of foundational work for this. And we're now back collaborating, which I'm really excited about. Jessica Ringo, Remy, Mary, and Martine are all here. No picture of Dimitri, but he's also here. So thank you.
Hello, thank you so much. I'm here. Hi, I'm Bam. I'm an assistant professor at NYU here. Thank you for the great talk. I have a sort of a conceptual question. So you've shown that the activation of the nerve endings elicits the immune response through the IL-1 beta. I was wondering, can you imagine that it's the other way around, or the sequence of events is that the immune activation could actually trigger the nerve sensitivity? And my second question is, in regards to the ATP release, as you know, there's also the clearance of ATP through the CD39 and 73 mechanism that will lead to the generation of ADP, adenosine, which can then dampen the inflammatory responses. Have you looked at these mechanisms in your system? Thank you.
Okay, so oh, I should go over here, I'm sorry. Yeah, so thanks for the questions, and we should chat afterwards too, but love to get your input. But, so number one, I'm trying to understand, so in terms of sequence of events, I think, we were actually able for the ATP, I didn't show you, but we used an analog of ATP and showed that we could bypass the channel and drive hypersensitivity. That doesn't really get at your questions like who comes first. And so, I don't want to, really shouldn't imply that it's, it's purely sequential, that one thing leads to another. But I think, that capsaicin is only depolarizing TRPV1 expressing neurons. I mean, there may be, maybe some other cells that we don't know about, but at least if one agrees with that assumption, which we can certainly have a debate about, but if the targets are the TRPV1 expressing neurons, then that's likely to come first. And certainly we don't know, but maybe other people do know that certainly we believe that the 2.2 channels are primarily expressed in neurons. At least there's pretty good evidence that is the case. And so, we believe that the 2.2 channel is on the neurons, and we've been able to do calcium imaging to show that the 2.2 channel drives calcium entry into the TRPV1 expressing neurons. And so we think we've got those two nailed down. But I think that your question is a really good one, which is, is there coincident activation of other release of other agents? I think that's completely and totally possible. So that's right. Second question was the ATP. No, honestly we haven't looked at that at all, and so it's again a really good question. We don't know, so we haven't looked at ATP clearance at all. Yeah. Brilliant. That's what I'm hoping. That's why I chose to talk about this as well, because I think that this is an area that we're very excited about. And we'd love to talk to people who are interested in collaborating. Yes.
Right here. Good morning.
Hi.
I was wondering somehow the same in terms of
the
ATP release. I see that you assume that all the ATP is released by vesicles, but there are actually other type of RV-NAT, connexins, pannexins as well.
Yeah.
And interestingly, pannexin and connexin 43 are activated by phosphorylation induced by calcium via SFK, family derived kinase.
Right.
So they can open and let ATP release, but also get calcium inside, generating also in other systems, especially in the spinal cord, positive feedback loops.
Yeah.
So my question is, have you considered other types of mechanisms of the ATP release? Because it seems like ATP release is essential to stimulate the immune system and get the IL-1β alpha.
Yeah. Also, thank you. No, we haven't. And, I've drawn this very, very naive kind of cartoon. And so we honestly don't know. We're very neuronal centric and, imagine that everything starts with a neuron, and that's almost certainly not true. And so, again, love to talk to you about that. Love to talk about other possibilities, of where, these pro-inflammatory mediators and secondary inflammatory, the origins of those. Because once we know that, then we can be able to do some additional manipulation. I think it would be super exciting. But thank you. We haven't looked. Yeah.
Wonderful. And just very quick question. Have you explored the possibility of actually having some purinergic receptors in the TRPV1 positive nociceptors? Because as far as I may recall, there might be P2X3.
Yeah, there definitely. So are the other subtypes, and we haven't explored those. We tried a few different things. There was, we always try our control experiment, and some of the blockers we used didn't, were affecting baseline, and so we didn't, so there's some non-specificity, but this particular antagonist actually worked brilliantly well, and so that was the one. But we haven't done deep exploration of the other receptors either, and so very important points. Thank you. Yeah.
Great. I think in the interest of time, we have to move on because we are a little bit late. But thank you for the great talk, and thanks for the connection to neuroimmunology, which is really a fun, very exciting field actually. Yeah. So thank you for that.